Hello and welcome to Basic Medical Sciences. So in this video, we're going to talk about glycolysis, right? So glycolysis is a metabolic pathway that converts glucose to pyruvic acid or pyruvate. The free energy released in this process is used to form high energy molecules of adenosine triphosphate, thus ATP, and reduced nicotinamide adenine dinucleotide or NADH. Glycolysis does not require oxygen, and it takes place in the cytosol. It consists of a sequence of 10 reactions which are catalyzed by enzymes. So for simplicity, I'm going to employ a new presentation to present like the intermediates of glycolysis. For example, if I want to represent glucose 6-phosphate, right? Glucose is a 6-carbon molecule. So I'll put them this way: carbon 1, 2, 3, 4, 5, and 6, right? Glucose 6-phosphate, right? So let's say this end is carbon 1 and this end is carbon 6. So glucose 6-phosphate will look like this, right? Okay. But what you need to remember, I will stress this out again, but just remember, glycolysis does not require oxygen and it takes place in the cytosol. Remember, this is very important, right? Okay, so let's say like this whole screen is a cell, right? A hepatocyte as an example, right? So glucose is taken into the cell through specific transporters like GLUT2, for example, right? GLUT2 is insulin independent, right? Okay, we will talk about GLUT transporters later, right? But for now, I just want you to understand this pathway, right? So this is glucose, right? So glucose is converted to glucose 6-phosphate, right? That is the first step. And this step is mediated by an enzyme known as hexokinase or glucokinase, right? So this is important. Glucokinase is the enzyme which is found in the liver and only in the liver, right? And hexokinase in other organs, for example, in muscles, right? Okay. In this process, you can see the first thing, check the colors, right? The arrows in black means these pathways or these reactions are irreversible, right? So you need to pay attention to that, right? So here, what we did here is like to put glucose into the cell, and if we leave it like that, it will just go out the same way it entered, right? So we need to put something so that it won't go out, right? So we'll put a phosphate. That's how it became glucose 6-phosphate. How do we do this? We use ATP, right? Simple. The second step is conversion of glucose 6-phosphate to fructose 6-phosphate. All right, so this one is more like a fructose, I told you, is a ketone, right? So it's just like we can say it's an isomer, right? In this case, it's an isomer. So the enzyme used is phosphohexose isomerase, right? It's easy, right? So fructose 6-phosphate is converted to fructose 1,6-bisphosphate, not biphosphate, right? Because biphosphate will be like this phosphate can be like close to one another. For example, this one is on carbon 6 and the next one will be on carbon 5, right? But they are separate, like they are in both ends here, bisphosphate, right? So this pathway is catalyzed by an enzyme known as phosphofructokinase 1, right? But look here, we've added another phosphate here, so it means we've used ATP again, right? And the enzyme, I said, is phosphofructokinase 1 or PFK1, right? Now, this fructose 1,6 bisphosphate is now cleaved by another enzyme called aldolase, right? So aldolase will cleave fructose 1,6 bisphosphate into these two molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, right? So these two, right? It's more like we just cleaved like at the center, leaving 3 carbons and a phosphate, 3 carbons and a phosphate, right? So this one is more like a ketone, it's a ketose, and this one is an aldehyde one, or an aldose, right? But this is what happens. Usually glyceraldehyde 3-phosphate is the one which we use like in this pathway. So we convert this molecule into this, meaning to say everything from this point will be times 2, right? Taking the place of this molecule, right? So here we have 2 glyceraldehyde 3-phosphate, right? There are 2, right? Now, glyceraldehyde 3-phosphate can be converted to 1,3-bisphosphoglycerate. Again, bisphosphoglycerate in both ends, right? We have added another phosphate. It means, okay, the first thing, we use glyceraldehyde 3-phosphate dehydrogenase for this pathway. And as I said, we use a phosphate. We have dropped a phosphate there. But for dehydrogenase, every time you hear about dehydrogenase enzyme, we talk about NAD positive, right? So here we formed NADH, right? So 2 NADH are actually considering, I said like everything from this point is times 2, right? So 1,3-bisphosphoglycerate is now converted to 3-phosphoglycerate, right? 3-phosphoglycerate, this one. But as I said, it's times 2, right? The enzyme for this pathway is phosphoglycerate kinase, right? Kinase, it means transfer of phosphates, right? So in this case, we use 2 ADP molecules to get ATP times 2, right? So this is the first step where we get ATP in this process, right? Previously, we just used ATP here and here, but now we have gained 2 here, right? So we can say it's balanced for now, right? So 3-phosphoglycerate is converted to 2-phosphoglycerate. It's more like a mutase because we have changed the position from position 3 to 2, right? So we used phosphoglycerate mutase, right? Then we convert again this 2-phosphoglycerate into an eno, right? So we use an enzyme called enolase to form PEP, or phosphoenolpyruvate, right? Phosphoenolpyruvate. This one is times 2, right? And then finally, PEP or phosphoenolpyruvate is converted to pyruvate, right? Because pyruvate is our end product of this pathway, right? And there are 2 again, and we use an enzyme called pyruvate kinase, right? So I want you to remember this pathway. The first step of glucokinase or hexokinase, and this step of phosphofructokinase 1, and then this last one, pyruvate kinase, right? It's very important, right? So there is another pathway. Okay, so here is a kinase. So we gained 2 ATPs, right? So we can say the net gain is 2 ATPs, because here we have balanced. Here we used the 2 and then we gained them here, right? So it's balanced. So the net is 2 ATP. In total, we have gained 4, but we used the 2 here. So the net is 2 ATPs, right? So there is this molecule called 1,3-bisphosphoglycerate. It can be converted into a molecule called 2,3-bisphosphoglycerate, right? So this one in hemoglobin, if you remember, you have this molecule. Like, so the conversion from 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate is through a bisphosphoglycerate mutase, right? So this one is very important. Like it stabilizes the hemoglobin, right? Like for like the form of hemoglobin in this case, it will have like low affinity for oxygen, and it's also known as Bohr effect, right? So we'll talk about it later. All right, so as a result, here I said the end product here is pyruvate, and actually we have two, right? So let's see the fate of pyruvate, right? So glycolysis here from glucose to pyruvate, right? So we have four pathways, right? So we will talk about the first one later. This one is called Kyle cycle, where we convert pyruvate to alanine, right? The second one is like from pyruvate to oxaloacetate, right? Using pyruvate carboxylase. This one we'll talk about it later when we talk about gluconeogenesis. I will explain it to you, right? But for now, you need to focus on these 2, 3, and 4, right? So the fate of pyruvate, it depends whether there is oxygen or there is no oxygen, because glycolysis firstly in the cytosol is anaerobic, but now if there is oxygen, then this pyruvate will enter into the mitochondria, right? Into mitochondria, where via a transition step it will be converted to a central metabolite called acetyl-CoA, right? Called acetyl-CoA, right? So the next video we will talk about the transition stage, this pathway, right? So this is what happens if there is oxygen, right? If there is no oxygen, pyruvate can be converted to lactate or lactic acid, right? So this one, this reaction is reversible, and this pathway is known as a Cori cycle, right? So the enzyme we use is lactate dehydrogenase, right? So you will see in most questions, if the person is exercising, right, and the energy or ATP is gained through anaerobic process, but will be accumulating this lactate. So this is what contributes to fatigue, right? Okay, so here on this side, I just it's just an explanation, right? So functions of different pyruvate metabolic pathways and their associated factors, right? So for the first one, pyruvate to alanine, right, or alanine is mediated by alanine aminotransferase, and this pathway need vitamin B6 or pyridoxine, right? So it carries what? It carries amino groups to the liver from the muscles, right? The second one, pyruvate carboxylase. This enzyme is a carboxylase. It means it needs biotin or vitamin B7, right? And oxaloacetate can replenish tricarboxylic acid cycle or be used in gluconeogenesis, as I said. The third one, pyruvate dehydrogenase, this enzyme, it uses like coenzymes. The mnemonic says the lovely coenzyme for NEDs. The lovely coenzymes for NEDs. The on T, that's in that order, it will be thiamine, that's vitamin B1. Lovely is lipoic acid. Coenzymes is coenzyme A. Coenzyme A is from vitamin B5, right? 4 is riboflavin, that's vitamin B2. NEDs is vitamin B3, right? That's niacin, right? So this one is actually a transition from glycolysis to tricarboxylic acid, also known as Krebs cycle, right? The fourth one here, lactic acid dehydrogenase, this enzyme, right? It needs vitamin B3, niacin, right? Is actually like the end of anaerobic glycolysis and is the major pathway in RBCs, white blood cells, kidney medulla, lens, the testes, and cornea, right? So it means like what I just told you, like these organs, like they don't have mitochondria, right? In RBCs, you don't have mitochondria. So this one is the major pathway, right? So in conclusion, right, I told you at first we have glucose, right? Glucose is 6 carbon, right? So we cleaved it into 2 pyruvate molecules, right? That's the first thing you gain. Secondly, we used these 2 ATPs, if you remember, and the gross, like in total what we got is 2, right? But we used 2, so the net will be 2 ATPs, but a total of 4 is what you gain, right? And also we've used 2 NAD positive, right? What you gain is 2 NADH, right? So this molecule will be sent to the electron transport chain, and we will talk about it later. Thank you so much. If you like this video, please make sure you subscribe so that you won't miss any of our latest videos. Until next time.